Okay, we're going to be talking about skeletal muscle, or not skeletal muscle, just the muscle tissue in general. So when talking about muscle types, there are three different muscle types that we want to focus on: skeletal muscle, smooth muscle, and cardiac muscle. We're going to touch on some of like the main ideas of it, like the things that kind of differentiate them from each other. We'll touch on those right now, but then we're going to kind of focus in on skeletal muscle, and we'll look at smooth muscle and cardiac muscle later on. So looking at specifically at skeletal muscle first, remember that skeletal muscle is attached to bone through tendons, and on the very outermost layer of the skeletal muscle is called the epimysium. You should notice this in gross lab. You can almost peel like this membrane off of the top, or there's like the fascia on top of the muscles that kind of hold like the whole muscle together, and that is that epimysium. Then you have these little chunk groups together inside of it, and that's surrounded by perimysium. And you have like the different vessels, the arteries, the veins, and the nerves running in between these fascicles surrounded by perimysium. And then we get down even further, and we have a muscle fiber, aka myocyte, aka muscle cell. When we're talking about the muscle cell, we're talking about a muscle fiber, and that is surrounded by the endomysium. Endo meaning most inside, myo meaning muscle. So we have that muscle fiber, and then within the muscle fiber, the muscle fiber is made up of myofibrils. These are rod-like units running the length of the muscle. So each of these muscle cells literally is running from one end of the muscle all the way to the other end. It's one continuous cell through the entire length of the muscle. And myofibril, even deeper than the myofibrils, we have the myofilaments, which is made up of actin and or myosin. They're two different things, but they kind of all join together. And we'll see that a little bit later on, exactly how that's all built up. So just another example, we have the muscle. We have that muscle fasciculus, the muscle fiber, the myofibril, and then we'll go into all of this different chunk. We have the sarcomere. Each of those different fibrils are made up of the actin and the myosin and some other proteins that we will look at. Okay, here's just another picture. The muscle, the fasciculus, and the myofiber, and then the myofibrils inside of it. Okay, so skeletal muscle. The important things to remember that they are elongated parallel cells. We really also want to remember that these are multinucleated. This is something that is different between them, between different types of muscles. So we want to understand that. These are striated, and they are voluntary. Also notice that the nuclei are peripherally located, so they're located on the sides of the cells. They're voluntary. This is the only muscle that we're going to talk about that is voluntary. Voluntary meaning that is ran and controlled by our somatic nervous system. And these muscles attach to the bones through those tendons. So our smooth muscle is involuntary, such as like the uterine lining, the stomach, the intestines, all that good, sort of good stuff. It is uninucleated, so single nucleus in each one. And these are capable of prolonged contraction, where you get a slow contraction. They're present in places like the blood vessels, the walls of the hollow visceral organs. Visceral literally means like the organs inside. So think the stomach, those intestines, all that sort of area, those are visceral organs. And these are not striated. So you want to remember involuntary. They are uninuclear compared to multinucleated. These slow, prolonged contraction, and they do not have striations. This is just a little note. We'll talk about something called peristalsis, and this is the movement of food and all that other stuff through the different organs. Now, cardiac muscle. We have cardiac muscle is also involuntary. We do not have to tell our heart to beat all the time, and part of that is thanks to autorhythmic cells. We have within the SA node and the AV node and Purkinje cells within the tissue. This is called the pacemaker tissue right down here. We'll get into exactly how they function later, but basically they have special leaky, they call them funny channels. They're a type of leaky channel that allows it to continuously in a steady rate reach up to threshold and develop an action potential, and then to contract to keep our heart beating rhythmically. So cardiac muscle is involuntary again. It is also striated like skeletal muscle. It can be uni or binucleate. They could have one or two nucleus in each cell. They're short branched fibers. They have this Y shape, so you can kind of see this. You'll see in lab, the lab exams, you might get a picture of one of these, and that's something that you'll want to be able to identify. You'll notice it by kind of that Y shape. And also, something very, very important to know is that there's something called an intercalated disc. So it's essentially, so here is my cardiac muscle cell, right? And it's going to connect onto another cardiac muscle cell, and so on and so forth. This region right here, I'm going to kind of blow it up so it's bigger. We have these two muscle cells connecting, and there is literally a gap between them. This allows for the flow of ions through it. So when we talked earlier about action potentials within the neuron, very similar thing is happening here, where we have different ions coming in, different ions going out, in order to cause action potential, to pass an action potential, to cause contraction, to cause movement. And once that first cell gets depolarized and gets that signal, this is how it passes that signal. It'll pass the depolarization signal to next muscle cell, next muscle cell, next muscle cell, and they will start to contract. And that's how the whole heart, it's all connected really quickly, so then the whole heart can beat rhythmically and together with the tissue around it. So it's actually kind of cool. The heart, if you actually look at the heart, on the top you have the atrium, the two atria, the bottom you have the ventricles. And the way that they actually pump, it's almost like it's being squeezed. It happens really, really quickly. So like from the atria, the top starts to squeeze first, like this. The top starts to squeeze, and it passes and it contracts lower. So it's almost like squeezing it down into the ventricles. And when the ventricles contract, when it gets it squeezes from the bottom and squeezes like this and contracts like this, starting from the bottom up, to push and to pump the blood. And it's thanks to these intercalated discs, and these specifically where these intercalated discs are, it's actually called the gap junction. I kind of misspoke a little bit there. Just to clarify, the intercalated disc is where they are connected. The space in between is called the gap junction, which allows the electrical signal to pass from cell to cell. Desmosomes is a protein that is used to kind of connect and hold the different muscle cells together. Okay. So just to clarify, intercalated discs are where they meet, then they connect cardiac cells. You have desmosomes, which are the proteins that link and hold everything together, and then the gap junctions, which allow the signal to pass through to allow for the actual proper contraction and passing that signal through the different muscle cells. Okay, so we looked at the overall kind of big picture anatomy of skeletal muscle. We're going to look a little bit into the micro anatomy of skeletal muscle, okay? So with skeletal muscle, we have blood and nerve supply. We saw in those pictures earlier, so you have arteries and veins and nerves running through the different musculature. Skeletal muscle has high vascularization, provides oxygen, supports the production of ATP, and removes the metabolic waste, such as the CO2, lactate, adenosine, etc. This is all a review, remember, from Biochemistry 1. Biochem 1, we learned all about those things. Also, the skeletal muscle must be able to be stimulated or excited. In order for the skeletal muscle to be excited, a nerve has to run to it and actually transfer that signal. So we get a signal for skeletal muscle. Remember, it's voluntary. So we get a signal from the brain. It's actually going to be in the frontal lobe. That's where our motor cortex is. It's going to send a signal down nerves through the spinal cord until it eventually gets to the proper level where it's supposed to go out. Our spinal nerve, and it pops out of the spine, goes all the way to the muscle tissue. And once it's at the muscle tissue, it's going to synapse on a neuromuscular junction, which we'll eventually talk about later on exactly how that works. But at that neuromuscular junction, we get a signal there, and it continues and passes along this electrical information, which causes the release of specific ions and stuff, which then causes our muscles to contract as they are supposed to. And when we talk about nerves, we talk about efferent and afferent nerves. So the efferent nerves, efferent means that they're going away from the central nervous system, away from the brain into the peripheral nervous system, which goes to the muscles. Afferent goes towards the CNS, towards the central nervous system, so back into the spinal cord, up to the brain. And that gives us our sensory information. Within the muscles specifically, we'll eventually talk about this later, but within the muscles, we have what are called muscle spindles, as well as Golgi tendon organs. It's getting a little bit ahead of us, ahead of what we've talked about right now, but eventually we'll cover all this a little bit later on down the road. These two things are what are called intrafusal fibers. And then we have extrafusal fibers, and these are our myocytes. All of the actual muscle tissue, the sarcomeres, all that sort of stuff, those are our extrafusal fibers. We'll get more into that way later. Kind of the idea of that, though, is that the efferent motor neurons go to the extrafusal fibers, and the afferent ones come from these intrafusal and the muscle spindles and the Golgi tendon organs. Okay, and there's a couple of other little details in there, but we will look at that later on as well. Okay, so kind of a lot in here, just kind of breaking this down and adding a little bit of information. So when we talk about the specific skeletal muscle fiber, right? The fiber itself, the muscle fiber, is the same thing as the myocyte or the muscle cell. And that is made up of the myofibrils. There's something called a sarcolemma, okay? The sarcolemma is this plasma membrane all the way around the myofibrils in the fascicle, okay? So it surrounds the myofibrils. And I kind of added all this extra information here. So the sarcolemma is the plasma membrane of the muscle cell and is surrounded by a basement membrane and endomysial connective tissue. The sarcolemma is an excitable membrane and shares many properties with the neural cell membrane. In other words, action potentials will be carried along the sarcolemma in order to activate the muscle when it gets the message from a nerve cell. The sarcolemma generally maintains the same function in muscle cells as the plasma membrane does in other eukaryote cells. It acts as a barrier between the extracellular and intracellular compartments, defining the individual muscle fiber from its surroundings. So the perimysium surrounds the group of fibers called the fascicles. The epimysium wraps the entire muscle and sheath of connective tissue. And the sarcolemma, a thin elastic membrane, covers the surface of each fiber. Okay. So surrounding those myofibrils, and each of these myofibrils are running parallel to one another. Remember, they run the entire length of the muscle. There's also something called the syncytium, which function as if the cells are fused. So syncytium is a single cell that contains multiple nuclei. In humans, these can be found in the skeletal muscles. These muscles are made up of fibers, which are long syncytia, something like that. Pronunciation is weird. Okay, let's talk about the sarcolemma. We also have the sarcoplasm. So sarcoplasm is like the cytoplasm of the muscle cells. So we talked about cytoplasm in like the regular cell where it holds all the stuff. The sarcoplasm plays that same role as the cytoplasm in the regular other cells. The sarcoplasm fills each muscle fiber and contains glycogen as well as several mitochondria. Remember, we need a lot of the mitochondria to produce ATP for the skeletal muscles to work. So skeletal muscle is a red color as a result of the degree of myoglobin and vascularity, which provides a short supply of oxygen. Myoglobin is like hemoglobin, myoglobin just being the oxygen transporter and holder in the skeletal muscle, whereas hemoglobin is the one in the blood. Okay. So you can see kind of here how we have this sarcoplasmic reticulum. The whole sarcolemma runs all around the outside, and then we have the sarcoplasmic reticulum inside. And these other structures called T-tubules. Okay. So some of these specialized structures, sarcoplasmic reticulum. This is going to store calcium. We'll see why calcium is important in a little bit. And has a similar function to the smooth endoplasmic reticulum in other cells. Now, the sarcoplasmic reticulum, as part of it, have what are called terminal cisternae, which are going to flow right along next to the T-tubules. T-tubules stands for transverse tubule, and that's because the transverse tubules literally run transversely through all of our sarcolemma, between that sarcoplasmic reticulum, in order to transfer and carry the electrical signal. So it allows that skeletal muscle action potential to travel from the sarcolemma to the inner muscle cells in order to activate the inner muscle cells and not just the outer ones. Okay. Another little note, we have T-tubules and we have terminal cisternae, so kind of like this right here, this tubule coming right through here. And these on the side, these parts of the sarcoplasmic reticulum are our terminal cisternae. Those three things together, so two terminal cisternae plus the T-tubule, is known as a triad. So if you ever hear that said, it's a triad. That's what it is, those three things. So now the myofibrils are arranged in sarcomeres, which gives the striated appearance and provides the ability for all cells to contract in the same direction. We'll get more into that, exactly what that process is and what the part of the sarcomeres are a little bit later. And I actually want to jump over here for a little bit and kind of go through some of this first. So here, looking again here at these structures, this is super, super important to understand these structures itself, okay? Within this picture, we can see the whole muscle and the fascicle, now the muscle fiber, and this is the whole muscle fiber. This whole tubing around is the sarcolemma. They're called invaginations, or these holes, these little tunnels running through it. Those are our T-tubules going down inside of it. In green here, these are our T-tubules on the inside. Then we have the sarcoplasmic reticulum, kind of coating, surrounding all around each of these individual fibrils. Remember, whole thing, fiber. The whole muscle fiber, myocyte, muscle cell. Myofibrils make up the muscle fiber. And the sarcolemma is kind of like, just like we say, it's the plasma membrane, just like a regular cell has the membrane. We think of a regular cell, we think of often like a circle, where we have a whole circle with all of our organelles and everything on inside. We have the cytoplasm inside, and then we have a plasma membrane surrounding the whole thing. That sarcolemma is that plasma membrane, but then it allows these little individual tunnels, which are the T-tubules, which run along the sarcoplasmic reticulum, the terminal cisternae of the sarcoplasmic reticulum, and the sarcoplasmic reticulum surrounds the myofibrils, and the myofibrils are made up of sarcomeres, and these sarcomeres are made up of myofilaments. It's kind of a whole process. I would recommend kind of doing this a couple times just to understand the flow of kind of like what's what, and write out all the AKAs for all of the different things. Okay. So we're getting a little bit deeper now. So we looked at the muscle cell itself, the muscle fiber. Now we're getting deeper, the myofibril, okay? The myofibril is this right here. And it's made up of sarcomeres, which have myofilaments. Part of what makes up those myofilaments are two structures called myosin and actin, are our big ones, okay? And actually, I'm going to jump over to this to go over all this. So, first off, actin. I thought I had myosin, actually. I'll do myosin first. So myosin, think of myosin as like a protein that builds up the myofibril, okay, or the myofilament. Myosin is made up of two heavy chains wound together like as a tail and two light chains. These light chains are here in the head, okay? Heavy chain tail, two light heads, chains, and these are all kind of wrapped together. In this head region, we have ATPase activity, meaning that ATP can be used and broken down and energy released at that spot, at the myosin head. That's as far as I want to go here. The myosin filament, you can see kind of how they all are here. They're in a group, in a bundle. It's called a thick filament. There's another one, there's a thin filament made up of actin. And we're going to get more into this in a little bit, but it's the little mini intro is that the myosin is going to use the energy of the ATP to help it bind. Make sure that I am very clear on this. It's going to use the energy of the ATP as part of a process where it's going to help pull the actin filament along and reset to continue to do this to cause contraction of the muscle cell. So the myosin is going to bind onto the actin, okay? And exactly how that happens, we'll get all into that. So going back to this, see we have the two heavy chains and four light chains, 2 light chains per myosin head. That rod-like tail. The myosin heads interact with the actin, and that has the myosin ATPase. Remember, the myosin head is where we get ATP to bind, and it breaks it down to use its energy. Then we have actin, okay? Also known as the thin filament, we have the actin filament. So actin has a couple of different parts that make it up, okay? We have a couple different names for stuff. Each of these individual little bead structures right here, we call them G-actin. You string a bunch of these beads together into a little smaller chain, and then we call that F-actin. Okay? Then we bind a whole bunch of the F-actins together to get us this whole long chain of actin. Okay? So G-actin is the smallest, F-actin is a little bit big, more is a bunch of G-actins put together, and then a bunch of F-actins put together is the full actin chain. There, let me see, make sure that I don't get too far ahead. Yeah, G-actin is that globular form, F-actin is a filamentous actin formed from G-actin. The actin is going to be tethered or linked to something called a Z disc. So what we're talking about, all of this, we're talking about that myofibril inside of a sarcomere. A Z disc separates each of these individual sarcomeres. So the actin is going to be tethered to a Z disc. We have another protein called tropomyosin. So there's our actin. Tropomyosin, make sure that I have this. There is going back a little bit to actin. On actin, they're going to have these sites called myosin binding sites. Myosin binding sites is on the actin. So on the actin, you have myosin binding sites. Okay? On the myosin, you have actin binding sites because the actin binds there. On the actin, myosin binds. Okay? Covering these active sites, so preventing the myosin from binding to the actin, is a protein called tropomyosin. See it right here? It's this long chain. It covers and blocks all of the myosin head binding sites that are on the actin. Then there's another protein bound to the tropomyosin called troponin. Troponin is made up of 3 different proteins. You have troponin C, troponin I, and troponin T. Troponin T binds to the tropomyosin, so it's attached to the tropomyosin. Troponin I binds to the actin. You'll see this later. Actin, a lot of times we'll talk about it being represented by the letter I. Don't know why, but that's the way it is. And also troponin C, which has a spot to bind to calcium. That's our tropomyosin, which wraps around the F-actin, blocks the myosin binding sites, and prevents the cross bridges formation at low calcium in the sarcoplasm. Okay, and we'll get to exactly why that is in a second. Okay. There is our actin filament. We talked about that. It looks like a repeat slide. That is a repeat slide. Okay. Before we talk about this exact, like what's happening here, we're going to go over the sarcomere itself. Okay. Honestly, we probably should have done the sarcomere first. Big key thing to remember, the sarcomere is the functional unit of the muscle cell. Meaning that without sarcomeres, our muscles would not function. Okay? These are going to be really, really important. So the sarcomere is this chunk of the myofibril within a muscle cell that are what are actually going to cause the contraction of the muscle cell. Here's where it all happens. And I'm going to show this one right here. So this whole structure is a sarcomere. Within the sarcomere, you have different components. So each of these sarcomeres, you have a Z disc and a Z disc right here. Z disc to Z disc is another sarcomere, and these sarcomeres line up all the way through an entire fibril along the entire length of the muscle cell. So it's a sarcomere. A little bit within that, we have what we call the A band. The A band, let me make sure that I wrote it up here so I wouldn't forget it. So the A band is the length of space with the thick filament and can't include actin. We have an I band, which is just where the actin is, an M line, and an H zone. And we'll go over each of these individually. So within our sarcomere, let me pull this up. Where's a better picture? Here we go. So the way that our sarcomere is built, so here's our Z disc to Z disc. We have myosin, also known as the thick filament. We have actin, also known as the thin filament. And we have another protein called titin or connectin that helps with elasticity to help the sarcomere return back to its original shape afterwards. Big ones we want to know are the thin filament and the thick filament. So these thick lines represent the thick filaments. These thin lines represent the thin filaments. Actually, he has one here. So, definitions. The Z disc itself is going to mark the edge of one sarcomere when the light filaments attach here. This is never going to go away, but they are going to move towards each other during concentric muscle contraction. During concentric muscle contraction, they're going to go together. The M line is a center line that the thick filaments connect and stabilizes the thick filaments. This will never change. Okay? The M line is always going to be kind of essentially in the center of that sarcomere. Titin, we talked about that. It keeps everything aligned and allows things to recoil back to its original shape. Then we have the I band, the A band, and the H zone. So when we get into talking about exactly how all these things move, the sarcomere as a whole is going to contract. And these thin filaments are going to get pulled inwards by the thick filaments, which is the contraction of the sarcomere. That means right here, this H zone, which is the area which just has thick filament, no thin filament involved, is going to get smaller and smaller and smaller and potentially disappear if it's maximum contraction. The A band right here is where all of the thick filament is, and that's never going to change size. The I band, there it is right here. The I band shows just where there is actin. So it's between the two different myosin, the two different thick filaments of the sarcomeres next to each other. And I think as both of these shorten, it's kind of hard to kind of picture this. It's like when both of these are contracting, like the Z disc is almost like staying right here, but this myosin thick filament is moving closer to both of these. So the I band is also shrinking. So during concentric contraction, the H zone shrinks and the I band shrinks. The A band stays the same. The Z discs are still there and the M line is still there. Okay? The way that all of this happens, the way that we get contraction of the sarcomere, is going to be something that we call the sliding filament mechanism, or the sliding filament theory. Okay? So, that tropomyosin is covering all of the actin binding sites. Then we're going to have an influx of calcium. Calcium comes and binds to the troponin complex, specifically the troponin C, and causes that protein to restructure, to change shape. When it changes shape, it pulls the tropomyosin off of the binding sites, the myosin binding sites. It's actually written wrong. This should be myosin binding site. I've noticed that before. When the tropomyosin gets removed and gets pulled off, the myosin head is then attracted to the myosin binding site on the actin, and goes and binds right to it. As part of this process, it's going to start, like this right here is our starting position. It's going to quickly bind, and then it's actually going to pull the actin filament along. So the myosin head binds and then pulls. And that's called our power stroke when that occurs. Now, the way that this gets reset, let me see if I included that picture. I did not. But remember we said that on the myosin heads, which are made up of, each individual head is made up of 2 of those light chains. And that has the ATPase on it. ATP is going to come and bind onto the myosin head, and when it breaks that phosphate bond, it gives it enough energy to reset. So when ATP binds, it binds to the myosin head. The myosin head is going to release off of the actin filament and kind of stretch and recock and reset, to then, bam, connect again and pull. ATP binds, it releases, resets, binds, and pulls, and so on. And that's what the sliding filament theory is, that this pulls and this filaments act to the myosin filament. The actin filament slides this way over the myosin. And that's why it says here that the filaments do not shorten, the sarcomere shortens. That was a little bit all over the place, just because there's not like a great order to really do this stuff in. Trust me, I've tried to reorganize this a couple of different times, because there's a lot of kind of concepts that just kind of they connect together in kind of all over the place. So hopefully that makes a little bit of sense, kind of the process of, first of all, the anatomy, how it's all put together, and then also how it occurs. We'll get into a little bit more, because the one thing that we did leave out was, where does this calcium come from? Okay. And that we will get into a little bit later when we talk about kind of how the action potential travels through the sarcolemma and how it's kind of activated and the calcium gets released from the sarcoplasmic reticulum. Okay, and we'll talk about exactly how that whole process occurs. Okay, thank you guys so much for watching. I hope you guys have a great day.